We'll now continue talking about processes in the cell, and this part is about cell communication or cell signaling. So it's important, as you know, for cells to communicate with each other. Whether an organism is single-celled or multicellular, there are reasons that you would need to communicate from one cell to the next via chemical signals. Epinephrine is an example of this, the fight or flight response. That's the epinephrine, the hormone, is a signaling molecule that generates a cascade of responses, as you might see in something like this. The gazelle is going to definitely need all the additional benefits of the response to epinephrine. The increased heart rate, increased breathing, increased ATP production, increased release of sugar or the glucose via the breakdown of glycogen from the liver. All of these things are signaled by the presence of the epinephrine in the receptors of the cells. Now, there are also single-celled organisms that communicate with each other for various reasons. And by studying microbes of various kinds, we get an idea of how that works and how it has worked throughout the evolution of life on Earth. So Saccharomyces cerevisiae yeast has two mating types. They're called A and alpha, and they locate each other via secreted factors. And there is a signal transduction pathway that converts the signals into that response. And these are a series of steps that the signal on the cell surface stimulates a series of intermolecular reactions or interactions inside the cell that lead to the response of the cell. Here we have the example of the two mating types of yeast, okay? And they're exchanging mating factors. Notice that they have a receptor. The A strain has a receptor for the factor from the alpha and vice versa. And when they come in contact with each other, that allows them to fuse together and create a cell that is a cross between or a hybrid between the A and the alpha cell. There are lots of similarities in various kinds of pathways, not only in microbes but also in all different types of multicellular organisms, which suggests that we have some of these signaling pathways are very ancient and ancestral in all organisms, beginning probably in prokaryotes and modified later in eukaryotes. We also see the same sort of signaling in bacteria that allows them to sense population density. Individual cells of bacteria don't do a whole lot of harm, but when they aggregate together, they can produce a stronger response or a stronger interaction with the host to lead to their greater success. In multicellular organisms, it is essential to communicate via chemical messengers from one part of the organism to another. There are various kinds of cell junctions, of course, in multicellular organisms like plants and animals that allow the cytoplasm of the adjacent cells to come in contact with each other. Localized signaling, then we may have direct contact or cell-to-cell recognition from one cell to the next via gap junctions in animal cells or plasmodesmata in plant cells. We can have cell-to-cell recognition where we have recognition factors on the surface of one cell and a receptor on the next, so that they can recognize each other. You see something like this in immune response of plasma cells to macrophages that have the antigens of the invading cell on their surface, and then there are specific receptors on the surface of the plasma cells to be stimulated to make antibodies. Lots of other things can happen in various kinds of organisms. We have local signals, we have nearby signals, we have long-distance signals of various kinds. The long-distance ones generally are called hormones, and the ability of a cell to respond to that signal depends on whether it has a specific receptor to that signal. Local signaling is often called paracrine signaling, and you see here that we've got the secretory vesicles being released from the secreting cell, and the target cells are relatively nearby. These molecules diffuse through extracellular fluid to come in contact with the nearby target cells. Synaptic signaling is a special kind of paracrine type signaling that occurs in a synaptic cleft between a neuron and the target cell, which may be another nerve cell, or it may be a muscle cell or some other kind of cell that responds to the neurotransmitter. The electrical signal that travels along the neuron stimulates the release of the neurotransmitter, which then diffuses across the synapse to the adjacent cell, which has the receptors for it. A lot of drugs work by inhibiting some of this or enhancing some of this release of neurotransmitters. And then, of course, the long-distance signaling by the endocrine system, the hormonal signaling that we have when the endocrine cell produces the hormone that is then secreted into the blood vessel, which can carry the hormone throughout the body, and it will bind specifically to the target cell wherever that target cell happens to be in the body. And it will not affect other cells that do not have the receptors for it. Now, there are 3 stages of cell signaling. These were first discovered by Earl Sutherland, and he was studying how the hormone epinephrine acts on cells, and his suggestion was that there were 3 processes: reception, transduction, and response. And so we'll talk about each one of these in turn. So here we have an example of what happens. We've got a receptor on the surface, on the membrane of a cell, and the signaling molecule fits into the receptor, causing a change in conformation, a change in shape. This, in turn, stimulates a series of relay molecules in a signal transduction pathway, oftentimes by making change in conformation that will bump into another one or bind to a second one. And then finally, the final molecule in the transduction pathway will activate the cellular response of whatever it happens to be. And we'll talk about some examples of these as we go on. Now, in reception, the signaling molecule is going to bind to the receptor, and that's going to cause it to change shape. The signal molecule is called a ligand, or a ligand, you sometimes hear it called, and the receptor is very specific, similar to what we saw in the active site of an enzyme for its substrate. When the receptor binds the ligand, then we see a shape change in the receptor, and that's oftentimes the initial transduction. And then a lot of these receptors are plasma membrane proteins, although there are some exceptions to that, as we'll see in a little bit. Water-soluble signal molecules are going to bind to those specific places on the proteins that are on the plasma membrane. And there are 3 main types of membrane receptors: G protein coupled receptors, receptor tyrosine kinases, and ion channel receptors. So we'll talk about each of these in turn. G protein coupled receptors, or GPCRs, are the most common kind of cell surface receptors, and these are receptors that work with a G protein. A G protein is a protein that is similar to ATP or ADP, but instead of adenine as the nucleotide, it has guanine in it. And basically, they act like a switch. If GDP, or guanosine diphosphate, is bound to the G protein, then the G protein is inactive, and then if it gets phosphorylated to GTP, then it activates the protein. So here we have a signal molecule, and notice that it's got a lot of alpha helices that go through the membrane, with the hydrophobic parts in the interior and the hydrophilic parts toward the outside and the inside. So we would have a binding site here, and then this part would probably interact with the G protein. So here we have a G protein coupled receptor, and when it's activated by the signaling molecule, by the ligand here, that causes the G protein to be phosphorylated. The GTP phosphorylates the GDP that's on the surface, and that activates the G protein to move toward the enzyme. Here's the inactive enzyme, and so when the GTP comes in contact with it, it activates the enzyme, and that phosphorylates the enzyme, which then sends the G protein back to its back nearer to the receptor to be used again. So that's going to activate whatever the response is in the cell is going to be catalyzed by this initial enzyme. There's another group called receptor tyrosine kinases, or RTKs, and these are membrane receptors that attach phosphates to tyrosine. Tyrosine is one of the amino acids that's found in proteins. These can trigger multiple pathways at one time because they have multiple sites that can be activated. there are a lot of cancers that are related to deficiencies or defects or mutations in the RTKs, in the signaling of the RTKs. So here we have the RTK receptor tyrosine kinases. These are inactive when they're usually occur in pairs. When the signaling molecules attach or the ligands attach to them, they move together to form a dimer. And then they're activated at that point, and then they can be fully activated by being phosphorylated by ATP, and that phosphorylation process activates these relay proteins that can stimulate the cellular response. And you can see that since you've got several binding sites here, you've got several responses that can be activated at the same time. This is going to increase the response and make it respond not only faster but also more efficiently for the cell. There are also ligand-gated ion channel receptors, and these act as a gate. The ligands act as a key to unlock a gate when the receptor changes shape. So when the signal molecule binds to the receptor, then that gate opens up to allow specific ions through the channel of the receptor. As we see here, the ligand comes in contact with the site on the gated channel receptor, and you see here the gate is closed. This causes the binding here of the ligand causes a conformation change in the protein, which opens up the gate, and that initiates the cellular response, which allows the ions to pass in. Once that response is finished, then the ligand can disconnect, and that will close the gate once again. This is very efficient, and it's used in a lot of kinds of cells like nerve cells, muscle cells, that need to have a sudden influx or outflow of particular kinds of ions. Now, there are also intracellular receptor proteins, and these are ones that are found in the cytoplasm or the nucleus of target cells rather than on the surface. Oftentimes you have hydrophobic messengers that can cross the membrane, and when that happens, they can come in contact with a receptor on the inside of the cell. These are things like steroid and thyroid hormones of animals. And then these can act as these activated complexes can act as transcription factors that turn on specific genes to make specific proteins as the response. So here we have testosterone in the extracellular fluid here and a receptor protein on the interior of the cell. Since testosterone is a steroid-based hormone, it can easily pass through the membrane and form a hormone receptor complex with that receptor protein. It can then move into the nucleus and act as a transcription factor to transcribe the DNA into messenger RNA, which can then stimulate the production of a new protein that is necessary for the response of that particular hormone. That makes sense if you think about changes that happen in bodies in response to hormones like testosterone. A new protein might be to signal the development of a thicker cartilage in the larynx of a male going through puberty, or the production of or the stimulation of hair follicles in different parts of the body, and so forth. Now, transduction, which is the final step in the process, these are molecular interactions that relay signals from receptors to the target molecules. There are usually multiple steps in this, and the good thing about this is that a multi-step pathway can amplify or increase the response in the cell. That provides a lot more opportunities for regulating the response. The pathways are mostly proteins, and basically the receptor activates another protein, which activates another, and so forth. It's just kind of like the dominoes falling until finally the response protein is activated. Oftentimes there's a shape change in the protein or some kind of movement that occurs as a result of its interaction with the previous one. A lot of times this involves phosphorylation and dephosphorylation. So when you have these kinases, protein kinases, they are going to transfer phosphates from ATP to a protein, and then that's going to cause a conformation change, and then it can be negated by a process called dephosphorylation. There are enzymes called protein phosphatases that remove the phosphates from the protein, and that's going to turn off or on the activities as required, turning them off and on or up and down as needed in the cell. So here we have the signaling molecule, comes in contact with a receptor, which activates a relay molecule. We have an inactive protein kinase that becomes activated by that, and by by being phosphorylated by ATP, and then it's just a cascade of these down the pathway to finally activate the protein that's going to provide the cellular response. So one molecule affects the next molecule, affects the next molecule, and so forth. There are also such molecules that are called second messengers. Oftentimes they're small molecules or ions that can spread throughout the cell by diffusion and participate in pathways initiated by the GPCRs and RTKs we talked about earlier. Common ones are cyclic AMP or CAMP and calcium ions. CAMP, cyclic AMP, remember, is adenosine monophosphate, and so it's very widely used as a second messenger in a lot of pathways. There's an enzyme called adenylyl cyclase, which converts ATP to CAMP in response to a signal. So here we see that adenylyl cyclase removes the pyrophosphate, the 2 phosphates there, and leaves us the 1 phosphate, which then forms an ester bond with the number 3 carbon in the ribose sugar part of the adenosine part of the molecule. Then the phosphodiesterase can actually remove that ester bond and turn CAMP back into regular AMP, which can then be rephosphorylated in various processes to produce ATP again. A lot of signal molecules trigger the formation of this, and then there are other parts of the pathways like G proteins and GPCRs and so forth. This can also be regulated by systems that inhibit the adenylyl cyclase, so it can actually slow down this process as needed. So here's our first messenger, such as epinephrine, activating the G protein, which activates the enzyme, in this case is adenylyl cyclase, which then changes ATP to CAMP, which acts as a second messenger for this protein kinase, which then stimulates the cellular response. You can see it gets pretty complicated sometimes. There are also second messengers like calcium ions, and that's very important because cells can regulate the concentration of calcium ions by various methods, often gated channels. Here we have some gated channels of calcium pumps, okay, that can be activated by the phosphorylation by ATP. So we can pump calcium ions into, for instance, the ER, pump them out of the cell or into the mitochondrion as needed. Pathways that lead to this involve a couple of different molecules called inositol triphosphate, or IP3, and diacylglycerol, or DAG, as additional second messengers. So here we see another response to the signaling messenger. A G protein activates an enzyme phospholipase, which changes PIP2 to DAG and IP3, which are second messengers. The IP3 is a second messenger in this case, which then acts as the ligand for an IP3 gated calcium channel, which then allows calcium ions to be pumped out of the ER into the cytoplasm, and then our calcium ion is a second messenger to activate several other proteins for the cellular response. The response is leads to regulating transcription or other cytoplasmic activities. Oftentimes they are transcription factors of various kinds. But ultimately there's going to be some kind of regulation of cell activities that's going to be caused by or produced by the signal transduction pathway. A lot of pathways regulate the synthesis of enzymes by turning genes on or off in the nucleus, and that means that they may function as a transcription factor. So here we have a growth factor receptor that is going to be, the transduction is going to be through a phosphorylation cascade that's going to activate a transcription factor that will transcribe the DNA into messenger RNA to produce a protein response to the activation. There are lots of other pathways that regulate activity of enzymes rather than their synthesis. Sometimes they deactivate enzymes or activate enzymes. They can also affect other behaviors of the cell, like changes in cell shape and so forth. Here we see the transduction cascade from the binding of epinephrine that is involved in actually the release of glycogen from the liver into and breaking it down to glucose to release into the bloodstream. This is a necessary thing, of course, in the fight or flight response, because you need to have extra glucose in the bloodstream so that your cells have more glucose to use for fuel to produce more ATP for your flight or your fight response. Another process that uses a lot of cell signaling pathways is the process of apoptosis. Apoptosis is the programmed or controlled cell suicide. And in this process, parts of the cell are cut up and packaged into vesicles that are then digested by other cells. And the process of apoptosis prevents the enzymes from leaking out and damaging neighboring cells, which could be a real danger. Here we have a normal cell and an apoptotic cell next to it. You should see the difference in appearance when you have the cell that's in the process of dying. So the main proteases or enzymes that break up this or that cause this process are called caspases. And there are a number of different things that can trigger apoptosis, including extracellular ligands, DNA damage in the nucleus, and sometimes protein misfolding in the ER can cause this. This is a process that developed early in animal evolution, and it's essential for development of animals. There are lots of times when you need to get rid of extra cells in the process of development as moving from one stage to the next. It's probably also involved in diseases like Parkinson's and Alzheimer's. And perhaps interference with apoptosis may be a contributing factor to some cancers. If you interfere with the pathway that signals apoptosis, then you can keep that cell that should have died alive, and the cell that should have died is going to be abnormal in some way, and that can lead to the development of cancer. Here we have an example of apoptosis in a developing organism. This is a salamander, I think, and the interdigital tissue is present there. It just looks like a paddle in this point, but then you have the cells that are undergoing apoptosis to end up with space between the digits, which is necessary for the proper functioning of the hand as the animal matures.